Hi, everyone. We'll wait just a minute for everybody to join in, and we'll go ahead and get started. All right, so we'll go ahead and get started. Good afternoon, everybody. I'm very excited to introduce to you two early career scientists. They're both from Professor Ann Tate's lab at Vanderbilt University, and so they're two of our own. And so we'll start with Dr. Stephanie Birnbaum. Stephanie got her Bachelor's of Science at Emory University, followed by a PhD here at Vanderbilt in Patrick Abbott's lab. And then she then obtained a United States Department of Agriculture NIFA fellowship, one of the very prestigious fellowships, to do her postdoctoral research with Ann. And she'll be telling us about evolution under chemical warfare and variation in combat strategies. As a reminder, we'll have our traditional Zoom reception at 4:15 with both of our speakers, so feel free to join. And we'll send the information through the chat. And then also for each speaker, we'll have questions at the end of each talk. And so as you see Stephanie's talk, if you have any questions, please put them in the Q&A and we'll address them at the end of her talk. And so without further ado, Stephanie, we'll look forward to your talk.
Awesome. Thanks, Antonis, for that introduction, and let me share with y'all my screen. Y'all should be able to see my presentation now. So yeah, thanks for allowing me to share a bit of my recent postdoc research here in the Tate Lab at Vanderbilt. So insects are incredibly diverse and are study organisms for biologists who investigate a range of really interesting evolutionary and ecological principles. But many of these insect pests are agricultural pests or disease vectors, and we care about their evolution with respect to food security and human health. A number of pesticides have been developed to combat these insect pests, but we know that the evolution of resistance to these compounds has been well documented in the field and is near inevitable. Resistance to pesticides is a growing threat to the sustainable control of insect pests, and to effectively address this problem, it's necessary to understand the diversity of underpinning mechanisms involved. Despite decades of pesticide application and research into evolved resistance, there are still open questions regarding the variability or convergence of resistance mechanisms between populations or towards different pesticide types. And understanding how insects evolve resistance, potential costs of this resistance, and potential convergence of resistance mechanisms is important for predictions of the spread of resistance and population dynamics of insect pests. So to begin, to experimentally investigate insect interactions with pesticides, I used the favorite model organism of the Tate Lab, which you'll hear about from both Nora and I today, Tribolium castaneum, or the red flower beetle. This insect is a worldwide stored grain pest and has become a model for studies in development, immunity, and pest biology. Important for my studies, it has a well-annotated genome, and there's been documented resistance to all types of pesticides used against it. As I mentioned, there are a variety of insecticides, and many of them have neurotoxic effects. Organophosphates are a widely used class of pesticides that inhibit acetylcholinesterase activity in insects, and exposure results in hyperexcitation of synapses, resulting in eventual death. Pyrethroids are a different class of neurotoxic pesticide that disrupt presynaptic sodium channel function and also disrupt other ion channels in the important sodium-potassium pump. Pyrethroid exposure also causes nervous system excitation and eventual death. And both of these classes of pesticides are currently widely used against a variety of disease vectors and agricultural pests, including Tribolium castaneum. To combat pesticides, insects employ a variety of resistance strategies. Target site modifications commonly evolve in resistant populations, and mutations in genes encoding acetylcholinesterase and sodium channels have been described in organophosphate and pyrethroid resistant populations, respectively. Insects can also employ metabolic changes with a variety of detoxification genes. There are many canonical detoxification genes that help to degrade, modify, and excrete toxins. Cytochrome P450s are a large group of broad substrate enzymes that help to degrade toxins, and GSTs, glutathione S transferases, and UGTs, UDP glucuronal transferases, are both large detox gene families that help to modify toxins to excrete them more easily. And I mention these gene families because they'll become important in the data later. And last, the insect cuticle plays an important role in pesticide resistance. The cuticle not only makes up the insect exoskeleton, but it and other components like chitin and the peritrophic membrane are also key parts of the insect gut. In cuticle, chitin, and peritrophic membrane modifications in the gut can reduce permeability of toxins in insects. So do different populations have similar changes in detoxification and cuticular genes in response to pesticide resistance? And do insects employ similar resistance mechanisms in response to different neurotoxic pesticides? To investigate evolved resistance mechanisms to different pesticides, I used an experimental evolution approach to select for resistance to both organophosphates and pyrethroids in different tribolium populations. I then used organismal fitness assays to investigate how evolved resistance impacts survival and development under pesticide treatments. And last, I used transcriptomic gene expression analyses to uncover gene expression changes associated with evolved resistance. And so specifically, we can ask a number of different questions about how populations vary in the effects of selection with different pesticides. Do populations evolve resistance to both organophosphates and pyrethroids in the lab? Are there developmental costs to evolved resistance, and are there differences between pesticides? Understanding the potential fitness costs of resistance is important for understanding how resistant mutations might persist and spread through a population. Do populations have similar gene expression changes in response to pesticide selection? And do resistant populations express different genes than ancestral ones in response to pesticide exposure, indicating some important candidate genes involved in resistance? We can also investigate the convergence of evolution of resistance to pesticides with different neurological targets. Are similar genes differentially expressed in response to organophosphate versus pyrethroid gene expression, or versus pyrethroid selection? Do populations have similar levels of convergence with organophosphate versus pyrethroid selection? And these are important questions when considering the ways in which organisms evolve resistance to toxins and for implementing effective pest and vector control strategies. And so to start with my experimental design, in the summer of 2019, I used 6 beetle populations maintained by the Tate Lab and reared individuals from each population under either control, organophosphate, or pyrethroid conditions. These populations had been maintained in the lab under control conditions for at least 2 years prior to this experiment. And for each population, I transferred individuals on their respective selection regimes for 8 generations to create the selection populations. And for the main experiment, I reared beetles from each of the 3 selection regimes under control conditions for 2 generations to remove effects of previous pesticide exposure. I then exposed 12-day-old F2 larvae from each population to control and pesticide oral diets, either a flour yeast mixture or that mixture plus pesticide. And so the control selected ancestral populations were exposed to control organophosphate and pyrethroid oral diets. And then the pesticide selected populations were exposed to control or their respective pesticide diet. I measured mortality and development for 3 weeks. And I also collected individuals 2 days after being placed on their oral diet treatments for use in RNA-seq analyses. And then for this talk, I'll focus on the 3 populations for which we have RNA-seq data. And so through this experimental design, I measured mortality, development, and gene expression, and we can quantify the effects of selection, pesticide treatment, and the interaction between selection and pesticide treatment. And so here you can see with selection regime being shown by solid or dashed lines, you can see a main effect of selection regime on mortality, but no difference in pesticide treatment. Next, here you can see a main effect of pesticide treatment with higher mortality under pesticide exposure, but no difference between selection regimes. You can also imagine that both selection and pesticide treatment have significant effects on mortality. And last here, you can see an interaction between selection and pesticide treatment, where pesticide-selected individuals respond differently than ancestral populations to pesticide treatment. And so throughout this talk, I'll be referring to these effects of selection, pesticide treatment, and interactions between the two. So getting to the data, we see that populations evolve resistance to organophosphates after 8 generations. And so here, looking at survival hazard ratios on the left, the effects of our experimental factors on the probability of dying are shown. And so overall, here you can see selected individuals have equal chances of survival compared to the ancestors. And not surprisingly, pesticide treatment overall increases the probability of dying. But here, as shown through the interaction between selection on organophosphates and organophosphate exposure, you can see there's a significant positive interaction, indicating that selected populations are more resistant to organophosphate treatment. And so next, looking at the effects of selection and pesticide treatment on development on the right, with again our experimental factors, you can see the effects on the probability to develop to adults. And so here with regime, you can see that organophosphate selection slightly reduces the probability to develop to adults compared to those on the control selection lines. You can see that organophosphate treatment exposure has a negative effect on development. Overall, however, selected populations develop more quickly than ancestral ones when exposed to organophosphates, indicating adaptation in selected populations. However, when we examine the data for each population separately, you can see that there's variation between populations with respect to the effects of selection on survival and development. And so here, the proportion of surviving beetles is shown on the top, and the proportion of adults is shown on the bottom. Organophosphate selected populations are shown with dashed lines, and organophosphate treatment is shown in purple. And then the populations are in different panels. And you can see that overall, DORS evolved slightly lower resistance than the other 2 populations. And there are variable costs to development with between the populations with organophosphate selection and exposure. And looking at interactions with pyrethroids, pyrethroid selected populations are shown in dashed lines, and pyrethroid treatment in green, and again, populations in different panels. And you can see here with survival on the top, that all populations evolve similar levels of resistance to pyrethroids. And then again, with development on the bottom, you can see that pyrethroid treatment delays development in the ancestral lines, but in contrast with organophosphate selection, we don't observe cost to development for pyrethroid selection. And so overall, evolved resistance and impacts on development are more variable between organophosphate selected populations compared to pyrethroid selected ones, where we saw similar levels of resistance and no cost to development from resistance. But do patterns of differential gene expression reflect the fitness variation seen between populations and between organophosphate and pyrethroid pesticides? And so here, using RNA-seq analyses, we can investigate how selection and pesticide treatment impact gene expression. Do populations vary in their gene expression responses to pesticide selection and exposure? By examining interactions between selection regime and pesticide treatment, we can ask if selected populations express different genes and ancestral ones in response to pesticide exposure. And we can also compare gene expression between organophosphate and pyrethroid selection to ask if there's convergence in the genes differentially expressed under each of the pesticide selections. And so first, we see that populations vary in the number of genes differentially expressed with respect to pesticide selection and treatment. And so here, the effects of selection regime and pesticide treatment and the interaction between selection and treatment are shown with different colors. And I've reminded you of the graphical keys just for each factor effect. And so the experimental factors are grouped by population on the x-axis and the number of differentially expressed genes either up or down or on the y. And we see that populations vary with respect to the number of genes differentially expressed with each experimental factor. And we also see that there's population variation between organophosphate selection and exposure and pyrethroid selection and exposure. And so looking at the main effect of pesticide treatment on gene expression and the overlap of genes differentially expressed with organophosphate and pyrethroid treatments, we see a fair amount of overlap between populations for each pesticide, indicating that pesticide exposure elicits common gene expression changes. And in fact, there are 3 canonical detox genes, 2 P450s and 1 UGT, that are commonly differentially expressed in all 3 populations and to both pesticides, indicating the broad functionality of these phase 1 and 2 detox genes in interacting with different pesticides. And when looking at the effect of organophosphate selection on gene expression, so genes whose expression differs between the ancestral and organophosphate selection regimes, we see that organophosphate selected populations have highly divergent gene expression with little overlap between coffee and Snavely and no overlap with Doris, which differentially expressed only 4 genes. And I can perform tests to look for enrichment of functional categories in the differentially expressed genes. And we see the importance of the cuticle for coffee, while Snavely is enriched for genes involved in oxidoreductase activity, which is a function of P450s and other detox genes. And so here, different detoxification genes are shown on the x-axis with expression changes on the y, and different populations shown in different colors. And you can see that in response to organophosphate selection, coffee and Snavely are differentially expressing different detoxification genes, including a number of P450s. While Doris differentially expressed only 2 genes putatively involved in organophosphate resistance, a sodium-potassium transporting ATPase gene and a gene involved in oxidative stress. And here, in support of the functional enrichment test, you can see that coffee in particular upregulates a large number of cuticular genes in response to organophosphate selection. And next, looking at the genes that interact between organophosphate selection and exposure, we see that populations selected for organophosphate resistance have few differentially expressed genes in common when they're exposed to organophosphates. We again see the importance of cuticular genes for coffee, but Snavely organophosphate selected larvae differentially express genes involved in serine type endopeptidase activity when they're exposed to organophosphates. And OP selected Doris differentially expressed just 1 unannotated gene in response to organophosphate exposure. And indeed, Snavely differentially express a number of serine proteases and serpinpeptidase inhibitors, which are associated with serine type endopeptidase activity and are important in development and immunity. And interestingly, these genes have also been implicated in synaptic function, which is interesting when thinking about putative organophosphate resistance mechanisms. So overall, we see that variation between populations in gene expression responses to organophosphate selection reflects the variation seen in evolved organophosphate resistance and developmental costs to resistance. What about pyrethroids? So in response to pyrethroid selection, we see greater overlap in the differential gene expression. But when looking at the functional enrichment categories, we see differences between the populations. Coffee again is showing the importance of the cuticle in pyrethroid resistance, but Doris here is differentially expressing many more genes in response to pyrethroid selection, and these are involved in monooxygenase activity. And Snavely here is enriched for functions involved in heme binding, and importantly, both monooxygenase activity and heme binding are associated with P450 function. And indeed, you can see here with detoxification genes on the x-axis, and again, log fold change on the y, with populations in different colors, you can see that overall populations commonly differentially express a number of P450s, UGTs, and GSTs in response to pyrethroid selection. And looking at genes that interact between pyrethroid selection and exposure, we again see that populations selected for pyrethroid resistance differentially express similar genes when exposed to pyrethroids. Coffee and Snavely share a large overlap with Doris, and both have genes involved in oxidative reductase activity, indicating the importance of P450s again. And Doris has many more genes interacting with pyrethroid selection and exposure, and these are involved in hydrolase activity, a broad detox function. And interestingly, in contrast to the cuticular gene expression we saw in coffee with the organophosphate selection and exposure, we see that pyrethroid-resistant Doris instead are upregulating a large number of cuticular genes in response to pyrethroid exposure. And so overall, we see that pyrethroid selection and exposure have more similar effects on gene expression between different populations, and this reflects the similar organismal patterns observed with pyrethroids. And last, we can think about the convergence of differential gene expression between organophosphate and pyrethroid selected populations. And so you can see here that populations vary in the overlap of genes differentially expressed with respect to selection on different pesticides. We see the greatest overlap between organophosphate and pyrethroid selection in cuticle in coffee, with the cuticle being important in resistance to both types of pesticides. We see less overlap in Snavely, but functional categories for both organophosphate and pyrethroid selection are associated with P450s. And Doris, interestingly, had really different responses to organophosphate and pyrethroid selection, with many more genes responding to selection on pyrethroids and being important in monooxygenase activity and P450s. Yet half of the genes involved in organophosphate selection were also differentially expressed with pyrethroid selection. And so to conclude, we see the evolution of resistance and costs associated with this resistance are dependent on population background and pesticide type. We saw that pesticide resistance evolves after 8 generations, but organophosphate resistance is more variable compared to pyrethroid resistance. There's a small cost to development with organophosphate selection, but this cost was variable between populations, and we saw no cost to development with pyrethroid resistance. We also saw that the convergence of resistance mechanisms between populations varies between pesticide type. Genes responding to organophosphate selection are highly divergent between populations, with enrichment for different functional categories between the populations. And in contrast, we saw greater convergence in gene expression between populations with pyrethroid selection, with many enriched functional categories related to P450s. And populations varied in their convergence of resistance towards different pesticides, with the most similarities being seen between coffee organophosphate and pyrethroid selection, and then really different responses happening with Doris in selection to the 2 different pesticides. So overall, this work highlights the importance of the consideration of these complicated factors of population variation and the convergence of resistance mechanisms when employing effective pest management and vector control strategies. And so last, just really quickly, I'd like to mention some ongoing work that I didn't have time to discuss today. We've also investigated the impact of pesticide selection and exposure on immune dynamics using oral and septic infection experiments, looking at life history effects and gene expression and effects on chronic infection. And we've also looked into other costs of fitness or other fitness costs of resistance using fecundity assays with the organophosphate populations. So I look forward to hopefully being able to share these results with y'all at some point in the future. And with that, I'd just like to thank the members of the Tate Lab, our leader, Ann Tate, and then Nora, who you'll hear from in a few minutes, really helped me out with a lot of the infection stuff. And Destiny has been working on the ongoing fecundity stuff. And then Carly is an undergrad that helped with some of the GO enrichment tests. And then last but not least, Sitchin has been maintaining all of these different pesticide resistant populations in the lab since I've been remote. So with that, I would like to thank y'all and I guess open it up for questions.
Great, thank you so much, Stephanie. Yes, if you have questions, please feel free to enter them through the Q&A box and I'll ask them. I have lots of questions. That was a really exciting talk and really interesting talk. So let me start with one. What are your thoughts about, have you characterized the variation in the ancestral population that you started with? And, what I'm trying to get at is, do you think these differences that you're seeing are genetic? Do you think they're epigenetic? What is your sense of the variation of the genetic basis of the variation that you're observing?
Yeah, well, based on my RNA-seq analyses, I can tell you that there's quite a bit of variation just overall in population gene expression. And so these populations had been collected from, one was generated from Carolina Biological Supply Company, but the rest had been collected from around the East Coast. And so they're from disparate locations and distinct times. It's interesting to think about how they might differ in their history with pesticide exposure in the field. And so we don't have a lot of information on what pesticides they were exposed to in the past, but that is something interesting to consider when thinking about variation. But I do think it's genetic, and especially since we reared out these populations under control conditions, I think a lot of what we're seeing are more genetic changes, not epigenetic.
Oh, that's really interesting. And so that leads me to my second question, which is, I imagine some of the, for some insect pests that have been exposed and have evolved resistance to organophosphates or pyrethroids, that the mutations have been characterized. And so I'm wondering whether you've looked at the overlap between the genes you see in your experimental evolution analysis and what is known from retrospective studies, so to speak, of how pests out there are evolving resistance.
Yeah, absolutely. So, I mean, we know that P450s and GSTs and esterases are involved, and specifically involved in resistance to OPs and pyrethroids. And when I look at these, especially the genes that are commonly differentially expressed, the specific P450s that I'm identifying have not yet been involved in resistance to these pesticides, nor has that UGT. So that is interesting. And I think particularly interesting are kind of these things that are not commonly described in these resistant populations. And so I'm really interested in that one gene that's really differentially expressed in Doris that's involved in oxidative stress. And it's a gene that is expressed in the brain. And so I think, that could be a really interesting candidate for organophosphate resistance that could be interesting to look at in other resistant insect species, for sure.
All right, wonderful. I don't see any other questions in the Q&A, and so we'll go ahead. Thank you again so much, Stephanie, for a wonderful talk. And so we'll switch and move to Dr. Nora Sultz.